Morning, Bradley.
Morning, how are you?
I'm doing pretty well, thank you.
Good. Yeah, I saw your email. I was going to respond. You had updated us on your latest.
Oh, yeah. I just send that out in case anybody wants to see it. I don't expect very many people to read it.
Oh, okay.
When I've been on the receiving end of such things, the first few are interesting, and then I think, I don't really want to hear any more of this. So, how are you doing?
Well, I'm doing pretty good. Not too much. Just staying safe and not doing too bad health-wise.
Good. Your friends and family are staying safe too?
Oh yeah, we're all doing pretty well.
It's kind of weird. The people swarming to the beaches here in California and Florida and things like that. It doesn't make sense to me.
Yeah. Yeah.
So what's on our agenda for today?
Well, we have, I think Dick is going to present something on his triangular life. I don't know exactly what that is. He'll tell us. And then I have a presentation at Virtual Worm on DevoWorm. I'm going to present it I'm going to kind of go through it in the session here today. So.
Okay. Well, I've got about 45 minutes. I'll have to jump out at quarter to 8.
Okay. Yeah.
Looking forward to finding out about triangular life.
Yeah.
Surprises me how many pentagonal lives there are on this planet.
Oh yeah. Yeah, 5 and 6.
I'm sure there's a good reason.
Yeah.
Yeah. Lots of flowers, starfish. Seems like bisymmetrical life makes a lot more sense.
Right. Well, I mean, I guess it depends on, I wonder if anyone's ever written a paper on that about why you would have, and then not just like free symmetry where you have all different varieties. It just kind of seems like you have a couple, you have 5-fold, 4-fold, maybe 2-fold. You don't really see 3-fold very much, but it's kind of.
Yeah. There are few flowers that have 3 petals, I think.
Yeah.
Yeah, like I guess clover. I don't know if that counts as a flower. It's more like leaves, but
right.
I kind of fizzled out on the presentations I was going to put together. Just don't seem to have the energy for it.
That's okay, yeah.
I kind of reckon in about 3 months I'm going to be feeling pretty chippy.
Oh, good. Yeah. Hello, Susan.
Good morning.
You're muted. Yeah. Can't hear now. No. Sometimes you have to go back out and come back in, like if you have your earphones, if you just plug them in. Oh, they're not in yet. I don't know if that'll make a difference. But. I still can't hear it now. Maybe we can use the chat if can't figure it out. But hi, Ujjwal.
Hello, Liza. How are you?
All right.
Hello.
Well, how are you?
Oh, nice meeting you. Nice.
Can't see your face, Dick, but
Oh, okay, I'll turn it off. That's strange. Hmm. You can't see my face, huh?
No.
not yet.
Supposed to be on.
Huh.
Okay, oh, I see.
There we go.
There you are. Oh, you're looking good, Dick.
Yeah.
That's fine. What? We went out and caught some fish yesterday.
Oh, good.
Oh, really? Where'd you go?
Oh, little roadside creek. Suckers.
Suckers. Well, why not? They're fun.
I don't know, they're big.
Oh, yeah. Some of those are just huge. They're great fun to catch.
Yeah, cleaning was not fun.
Cleaning them. Oh, you're eating them?
Of course.
Well, good for you. My recollection is they taste fine, but they're bony.
Now we're gonna have, Natalie's gonna make a filter.
Oh, all right. So you eat them bones and all.
Ground up. No, they're called mullet here. I don't know why.
They're called mullet. Interesting. Okay.
Okay, what's the agenda today, Bradley?
Okay. Well, I have a talk I want to go through that I'm giving on Wednesday to a group called Virtual One. And then
Okay.
I didn't know you had said you might present on triangular life.
Yeah, I'll show some pictures. Yep.
Okay. So, okay. Yeah, Susan's sound is not is inoperative, so she'll be chatting in the chat box. So yeah, Dick? Why don't you start with your pictures?
What?
Oh, you could start with your pictures, and then we'll go on.
Oh, you want me to start with pictures?
They're up.
Oh, I gotta remember. How do I share screen on this thing?
That's at the lower right-hand corner of the screen. If you go down, present now.
Oh, present now. Okay.
Yeah.
Present now. There we go. Okay, now select screen. Okay, select screen. I think it's 2. Let's see.
Okay.
Okay, I don't know what you see.
It says writing. DFCD.
What on earth does that mean?
I don't know.
You don't see my screen.
Oh, I see.
Guess
I see
not.
a screen, but it's like it's white and it says writing. It could be one of your windows.
Oh, shoot. No, I don't have a white window. Oh, wait. Maybe? No? I picked the wrong one. It doesn't show you the screen.
Oh.
So I'll stop the presentation, and I'll start it again. You want to present, and we still don't see anything.
Same thing.
How do I start over? Get a new screen.
You go down to the same place you picked before, and you can like cancel out of that. I think if you go down to the lower right, yeah, stop.
Okay.
And then,
let's see. Present now. Let's see what happens. Your entire screen. Oh, there we go. Okay. Now select the window. It gives me 100 selections.
oh,
I don't know which one. How am I supposed to know which one?
oh, I don't know.
I'll try it.
Yeah.
You don't really have 100 windows open, do you?
Maybe I do. Okay. Dismiss. I'll try it again. Yeah, this is ridiculous.
Yeah. Yeah, it should be the entire screen.
I have to grant permissions now.
Oh.
Okay, your entire screen. I get your entire screen. And I can't share your screen.
Huh.
You must grant permissions, and then it just says dismiss. Doesn't allow me to grant permissions.
Huh.
Ridiculous.
I don't know what to tell you.
Present now. Okay, a window. I'll pick a window. Can't share your screen. You must grant permissions. Oh well.
Okay.
This
Well.
is ridiculous. I think I can log out and start over.
Okay. Anyways, yeah, I don't know why it's not letting him do that, but we'll do that, and then I'll just go through the presentation, and it won't take that long, but oh, there's Vinay. Hello, Vinay. There's Dick again. I just wanted to mention. Hi. Before we start, the GSOC selections will be today, later on, so those will be announced when they're made. Or, like, we have to wait until, I can't remember, 1800 UTC to make the announcement, so we'll be announcing. Are you frozen, Dick? Oh, there you are. And I can't hear you. Yeah. There you go.
Very nice and here. Okay.
Yeah.
Now, will you allow to see your screen? Select window or screen. Now, I picked Untitled, Untitled 2. That leaves Untitled 3 to Untitled 15. Ridiculous. Let me try one here.
All right. All right. We've got a director. Yeah.
What is that? That's the one of the screens I got open. Oh, I see. Put the window. What the hell? Yeah, I might have the whole windows open, but
All right.
I don't know. Okay, closing lots of windows.
All right.
This is ridiculous. I had no idea when they opened. Too many options.
Yeah.
Oh, I finally found the window that claims I'm presenting.
Oh, okay.
But that's not the one I want to show you.
No.
Well, let's see. Well, maybe I can use it anyway. I don't know.
Okay, try it.
Let's see. Go here. Maybe. Let's try this one. There it is. Okay. There. You see, what do you see?
A file directory.
You see a file directory.
Yeah. Oh,
Now
now
what
it's
do you
changing a little bit.
Now do you see a paper?
No.
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 Oh, God. This is ridiculous. Okay. Can't. Yeah, I think you should go to Skype, but
Okay.
okay, let's stop presenting, stop screen sharing. Now I gotta figure out. Okay, why don't you go ahead and
Yeah,
I'll try to figure
I'll
this
go
out.
ahead. So yeah, let me present my stuff first, and then Dick can like reduce it.
I'm gonna close everything except this.
All right. So I'm going to present my screen now. And let's see.
What's the name of your screen?
Oh, I just share my entire screen, and I can just share exactly what you're seeing.
Oh, see, I have 3 screens, but I don't know what their names are.
Oh, okay. I don't know. Because I think you have multiple monitors, so.
Yeah, I have two side monitors,
Okay.
but I have no idea what their names are.
Yeah. So why don't I present? So let me do the presentation format here. Present. So the first thing I wanted to mention here is that today is May the 4th. So it's May the 4th be with you. It's Star Wars Day. And so, yeah, so people like to keep things online for this. And I found something kind of interesting, if maybe not useful, but interesting. Star Wars and spreadsheets. So this is where people have made art in Excel spreadsheets, and in this case, it's different images from Star Wars. So each of these, it's like pixel art, but each of the pixels is a cell in Excel, and they've changed the value to some color, and so they've been able to make these different graphics with that type of methodology. And then this is an API for generating like a database for different things in the Star Wars universe, and that's executed in Excel. Anyways, it's or actually in Google Sheets. Anyways, May the 4th be with you. So, so this is the talk I'm giving to this group on Wednesday. It's a group that's online. They started these online talks since everyone's been quarantined, and they've had some pretty good talks. They have them on YouTube. I think I sent out a link in the weekly email. They have like about 2 talks a week. It's an hour long on Wednesdays. And so this one is the, they call it the virtual worm, so I feel kind of at home with that, given what I'm presenting on here. So this is developing the virtual worm. So this is a data theoretical synthesis. This is basically a lot of stuff we've been doing in the group here. So the first slide is talking about the DevoWorm group, talking about the linkages with OpenWorm and with using secondary and tertiary data, and then the actual people doing the work or the topics that we discuss. And then talking about the sources of data, so people will be interested in where the data come from. So there's mostly microscopy data, maybe some molecular data that's never really been realized, but and then I would actually talk about some primary data later on in the talk. Then this goes into a sort of a pipeline of either segmentation and modeling, and that's the DevoWorm AI ML pipeline, which we've talked about. We also have cell tracking data, which is where you're tracking different cells in an embryo or in an image. And you can either use that to model and simulate or to analyze quantitatively. And then this is what we call DevoZoo, where we have all of our data sets and we can extract data from that. And then both of those can be used for analysis and visualization. And then I'm going to bring this up because a lot of these people aren't computational people. This quote by John von Neumann, with 4 parameters I can fit an elephant, and with 5 I can make him wiggle his trunk. And so what that refers to is this idea that you can take a bivariate graph, say, draw an elephant on it, given the right equation, and then you can actually animate it with additional equations. The point of this parable is that you can simulate something that looks like an elephant, but it isn't. It may not reflect your something real in the world. So you have this problem in modeling where you can model something that looks pretty good, but it's actually not what you want. You can recreate a lot of things with just a few parameters. Which actually stands in contrast to a lot of the deep learning stuff we've talked about, which is where they're using millions and millions of parameters to get answers that reflect the real world. And then to remind people again that you want to avoid spherical cows. So this is something that Dick's talked about, and I have an animation here of a cow going from a sphere to a cow. And so that's a reminder that we're working like from an embryo to a asymmetric organism, and so that's again relevant to this talk. And so I'm going to kind of go into some recent attempts at synthetic embryology. So people have actually attempted to model embryos synthetically, and I'm reviewing this because I want to give people an idea of sort of what it means to be sort of artificial, as what an artificial embryo might look like. So there are examples from biology where they've cultured artificial human embryos, a set of progenitor and organizer cells, and they call these things synthetic embryos. And so they can actually observe a lot of different aspects of molecular biology in these systems. And so there's a paper here from Nature on that topic. But then also, this is actually from a paper that Steve brought up to my attention. And this is something called the digital embryo. And so this is something where you have an organism that has this sort of development like C. elegans. It's the mosaic development. And they're able to track the cells in early development. They do the single cell sequencing. And they're able to map that data to a virtual model of the embryo. So here's the embryo, and they can map molecular data. So we're kind of doing something like that in a way, but we also have a problem of representation. So you have all of these layers of representation that we can use to model an embryo, and it's pretty complex.
Bradley, how many of these are model dependent?
Which ones?
Well, like the one you had about WNT.
Oh.
The paper.
Yeah.
Self-regulation by combined signaling. Now, is that all model dependent? Or are they just observing the distribution of these molecules?
I think they're just looking at the distribution of the molecules. They may be model dependent, I'm not sure. But it's not.
Yeah, I think that's very important. I mean, you got a problem here in that what you observe is driven by what you think that you're seeing.
Okay, yeah.
In other words, that you look first and then you look at the organism.
Yeah.
And you look at it through the eyes of the model.
Right.
It's a good way to go astray.
Yeah. Okay. So, anyways, I was going to talk about this problem of representation. So you have everything from genetic to behavioral representation. And so if you have these elements in your model, you can, you don't have to have every element in the model. You can only use it. What was that?
Which level involves physics?
Physics? I guess the phenotype, actually they all sort of involve physics, but maybe the phenotype and behavior are most physical, I guess. That's what I was going to point out. So you can represent the entire thing from genetics to behavior, but you don't really need to do that. You just need to represent a subset of those, and you can get a pretty good approximation. The question is, what mix should you choose? And so then I claim that, well, we're focusing mostly on phenotypic and behavioral representation, and those are the most physical as opposed to molecular and genetic, but those can be physical as well, depending on if you're looking at like distributions of molecules in space or gene expression or whatever. So we're going to focus on these two levels of representation. And so then I get into the 5-dimensional data structure, which is basically the way we characterize the lineage tree. So we have 3 dimensions of space. So we have these cell centroids in space, which are generated by cell tracking, and then those are boiled down to a series of cells in the embryo. So this is like a bunch of replicates, and you do a smoothing procedure to find the cell positions. But then you also have 2 additional parameters, and those are lineage time, which is this T value, which goes from a single cell to multiple cells through cell divisions. And this I, which can represent a number of things. It could be along the anterior-posterior axis. It could be the position in the lineage tree. There are a bunch of things it could be, but that's another 5th parameter. And so in this case, we have, call it context, but like I said, it's a pretty general parameter. This is an example, a 2D array of cartoon cells. So you have 3D space, and then you have time and context. And so that's the basic idea of how we approach this. So here's a paper that was done. Of course, this is from Dick and I on comparing C. elegans with Ciona intestinalis, and this is the tunicate or sea squirt. And this was actually the model that I showed of the morphoseq was the Ciona. It was a tunicate, just like this. So early development, you have this ball of cells, and it's pretty deterministic in terms of its cell divisions. And then it turns into this thing here, which has a tail and a head, and then ultimately it becomes this. It looks almost like a vase that sits on the bottom of the ocean as an adult. But it goes through several life history stages, is the important part. But we're really comparing the early embryo of the 2 organisms. So, and then these 2 organisms are right here. This is the adult tunicate, and this is the adult C. elegans. And each of those organisms represent mosaic development, which is this idea that developmental cells have a predetermined fate. And so they exist, C. elegans and Ciona and other tunicates are mostly of this type, although they do exhibit some aspects of what we call regulative development, which are represented also by fishes and mammals. And so fishes and mammals are much more regulative. And the difference, of course, is that the cell fate is predetermined in mosaic development. So you start with the single cell, and you go to multiple cells, and the fate can be traced through this lineage tree. And it's always the same every lineage tree you encounter, every individual you encounter, it's the same. As opposed to regulative development, where when you get this sort of division of cells in an embryo, every cell can be different. So you can't cleanly trace the lineage tree in any one way. You can trace the lineage tree, but the ordering of the cells is different. If you go back and you look at the position of the cells, they don't look, the cells, say, in the anterior part of the embryo might be a variety of different fates instead of like an emerging head. And so this is something I have to keep in mind. This study focuses on mosaic development. So this is where we're comparing, yeah?
Bradley, there are mosaic embryos that become regulative adults.
Okay.
And you have to watch out for that.
All right.
I think snails are an example.
Okay. I'll just make a note of that. So, all right. So this is an example of one of the analyses we did. This is cell volume versus division time in minutes. And this is where, of course, is comparing the two organisms at, within 100 cells of the single cell state. So we have a bunch of cells, and basically we're showing that there's a decrease in cell volume for division time in minutes. Although Siona shows some interesting patterns, it's a little bit different curve fit, and some of the reasons for that have to do with the way the Siona embryo divides. It's also notable that the Siona embryo is a fourfold symmetry. So the way the Siona embryo works is you get 4 founder cells, and then each of those 4 cells represent sort of a pole or a symmetry. So those 4 parts sort of divide. There's somewhat symmetrical to each other, but there's some differences in the lineage tree. So that's why perhaps you see this difference. But you can also discover new relationships in developmental cell lineages. So this is something we discovered in this paper that was not clear why this is going on, but we were able to divide up this cell volume versus lifetime of cell relationship into 2 groups. You have this group that fits to this trend line here, where if this lifetime of the cell is longer, the cell volume decreases. But you also have these outliers here, which kind of fit along this curve, although it's pretty weak. But you do have, they're all basically, most of them are not the non-AB lineage. So the non-AB sublineage is in the posterior part of the embryo, and these are cells that are going to go on to form some of the organs like muscle and gut, as opposed to the AB lineage. There's one here that's ABAR, but you can see it's maybe not close to this line. So there's something going on here that might be of interest for further investigation. And also, you see the same thing kind of in Siona, where you have these cells that are outliers out in this part of the distribution. So they're longer-lived cells. Their cell volume, this is log cell volume, they're longer-lived cells in general. So you have this tail of longer-lived cells in Siona. Again, not really sure what that represents, but that's something you can pull out with a quantitative analysis, or a quantitative comparative analysis. So, but we can also build towards a meta-embryo. So this gets back to what I was talking about earlier, why I kind of took that digression into this business about synthetic embryos. And so we have this type of data, like I showed you, the centroid data from C. elegans, but we also have it for the fruit fly, Drosophila, and for zebrafish. And so these are examples of this embryos for each of these. These are the types of point representations we can build. And so we have data for a number of time points. These data are taken from SSBD, which is a database run out of Riken in Japan. It's Ken Ho, who's actually been part of our group on and off. And they have a lot of different data sets with cell tracking data. So we can do this potentially in a number of organisms. But we are interested kind of in building this representation of a meta-embryo. And so one of the other things we've done in the group is to look at the distribution of major events. So we have this, I created this map for a paper. This kind of maps out a lot of the main events in the embryo, going through from fertilization to the connectome, through some of the behavioral milestones. And you can see that it goes to hatch. And this kind of gives us a baseline for kind of what the cells are doing as they're dividing and differentiating. So we've done things along this timeline in terms of characterizing the embryo quantitatively. So we've looked at cell, like sort of embryo networks. So we've looked at the proximity of cells related to sort of the shape that the embryo takes over time. We've looked at the early connectome, and so this is 300 minutes post-fertilization. These two figures come from two papers in Biosystems where you can get there's a lot more information about these methods. And then this is what the worm looks like at hatch. So this is the larval worm. So as we can characterize these, we can get a handle on, how we might characterize an embryo or how we might characterize development as sort of this more abstract thing. And we can also look at the distribution of cells as they differentiate. So these are all the families of cells, and their emergence in time and their number. So you have different families that emerge in time, their number, and their distribution over time. So you can see there's some families that, like hypodermal cells, which proliferate early, and intestinal cells, and then there are other cells that are more specialized that proliferate late. And you can even compare these distributions in histograms, or we can look at different classes of neuron, interneuron. We have those data available. And then finally, you can simulate the timing of divisions and differentiations using a mathematical model. So this is something that I've been working on with someone who doesn't come to the group meetings, but we've been thinking about this. This is my contribution to this. He's been working on some other parts of it. So this is using sort of the math of Poisson processes to understand like differentiation events and developmental time. So looking at how they might be distributed in time and how the lineage tree might be, how it might, it's not a regular set of divisions, it's not deterministic, but to try to simulate some sort of Poisson process that would represent this. Let's see, let me see what we have in the chat here. We got a message in the chat. Did we do directions of cell divisions? Not really. I didn't talk about that, but I didn't talk about it. I mean, I guess we don't really get at that directly in some of the work. Let's see. So I'm going to have to speed this up like when I give it, because there's too much in here, but I'm going to probably trim some of it. So then I talked about this information isometry method. So this is a method that we worked on a couple of years ago, where given two regulative lineage trees or developmental cell lineages based on different criterion. So if you have two trees that are based on different criteria, how do we assess the information of the developmental process? And so this is a reminder that developmental cell lineages contain multiple sources of information, and that you can compare equivalent lineage tree topologies using visualization and information theory. So that's why we look at these two trees by comparing them using a Hamming distance, which is like an absolute distance here between the trees. So this is an example of we're comparing two trees and we're putting the values on this tree. So in this case, our positions are one position away for each of the two cell cases, and maybe from 0 to 3 positions away. Actually, 1 to 4 positions away for each of these. So these are the binary identities of the nodes at the end of this tree. and these are the distances. So you can see how that plays out in a tree. But we can also plot this on a bivariate graph. So we can use this, like this chart, to show how we can draw lines between, say, like this one cell node and the two cell nodes here. And we can draw arrows to show the relationships, and then these dots represent distances. So we can look at the distance between two trees in that way. And so this is a bivariate graph of all the nodes sort of arrayed in this type of isometric graph organization. And each of these dots, these colors, represents a different Hamming distance. So you can see there's variety across the terminal nodes here of the tree. In this case, this is I think 120 or 108 cell or something like that, because 128 cell doesn't evenly divide. So you have different subtrees, it looks like, with different Hamming distances. It's almost as if your two different criterion that you're using are swapping subtrees, and so you have these distances of, a Hamming distance of 5, a Hamming distance of 3, and so forth. So it gives people a sense of, like, if you're using two different criterion to build a lineage tree, what the difference is between those. And then this is an example of sort of benchmarking this against random trees. So if I just generate a bunch of trees randomly and I compare it to C. elegans, C. elegans has a higher level, higher amount of information if I compare the C. elegans tree with any set of random trees. The C. elegans tree has much more information in it than the random trees. And so you can see this is sort of, if you think about just random, this is, random signal, and then this is actually information in the biological tree. So maybe there's another application to it, is looking at sort of the information in that developmental process. And then, of course, you can look at subtrees, so you can look at the different subtrees by themselves to see what the Hamming distances are between them. And so there are a lot of options for analysis as well. And so, but of course, this only works for bilateral symmetry very well. So for cases where you have fourfold symmetry, maybe we may think of a more advanced solution to this. And this is one of the things that has been not going very far up till recently, but is a potential new direction for this, which is using these isometric networks and better measures of information. So things like mutual information, where you represent the symmetry on these trees or on these networks, and then use a different type of information theory to get the information out of it. And so then finally, I'm going to talk about this. This is some empirical science that was done by me in a postdoc that I did. And this is where we're taking some data and modeling it to these curves. And so the idea behind these experiments was these are changes in fecundity in a wild type versus a mutant. Well, we have wild types and we have mutants. And this graph represents sort of the measurement of the worm without starvation and the measurement of the worm with starvation. And so the idea was that the worms were starved in a certain period of larval development called L1. And if you starve them in L1, they can survive for long periods of time without food. But it has a consequence on their future development. And so that's what we're trying to get in this set of analyses, is that we starve them in development, larval development. How many offspring do they produce in over 5 days, which is the length of their reproductive life? So we measured for 5 days. So this is an example of where you have sort of this control, which is where you have the L1 worm, which is a larval worm, and then you just let it grow to the adult and you let it reproduce, and then you end up with a reproductive curve, which is this blue curve here. By contrast, this orange curve is the curve that happens when you take an L1 larval worm and you starve it. You put it in media, like a buffer media, and you let it sit in like 15 degrees for about 5 days. and then you replate them and you let them grow to adulthood, and you measure how many offspring they produce. And so, as you can see, there's a decrease both in the peak number of offspring and the timing of their reproduction. So this is the difference in N2, which is the wild type worm. So there's no genetic mutation here, no defined mutation. And this is one of the things you can do with these data. So there's a difference here clearly, but what we want to know is like what are the differences between different time points. And so in this case, we can look at the area under the curve, or rather, the differential area under the curve. So this is where we have one curve that's bigger than the other, but we don't know how much it is. So, but we can use this differential AUC measurement. We can use this. We can represent both of these distributions in a geometric space, and then calculate the size of this overlap, or the size of the overlap, and then the size of the curve that's bigger, and subtract the overlap, and you end up with these numbers. So this is so early on, the unstarved wild type is reproducing a lot more. It has a peak at about 2 days, and then it declines to very few offspring later in its life. And so there's a positive amount of reproduction for the one that's not starved. But the one that's starved, it's getting a little bit later start. So it actually doesn't do much reproduction until day 3, where then it's reproducing more than the one that wasn't starved. And so we have this measurement here. So we have these numbers to attach to this process. And then we can look again at the different. So we have 3 different components here we can look at. And then there's a lot of data in this bioarchive paper if you want to know more. But there are 3 different things going on here. So there's a shift in a skew in the distributions. So this is a shift in the peak reproduction to a later point of adulthood. So when you starve them, you get the skew in the distribution, which pushes it back 1 day, the peak reproductive period. You also get delay, so there's a delay in sort of where little to no reproduction occurs. So you can see these points 1 and these are sampled at every daily point. So from day 1, there's no reproduction at all. Day 2, there's very little, and then you get this. You go right to the peak at day 3. So you can refer to this as a delay in reproduction. So they're not putting their eggs out for another day after they've been starved. And you can also then also look at the kurtosis, which is if you look at the peak reproduction, what's the change? So in this case, you have a reduction of the peak, and that's in statistics they call it kurtosis. We can call it kurtosis. It just means that the peak value is lower. So that's all well and good for wild types, but what about for other defined mutants? And so in this case, we looked at 3 different defined mutants, and they're all this AAK 1 and 2 variety. So AAK is a gene that controls metabolism. And so if you starve, in the literature, they suggest that if you starve AAK mutants, they become they have deficits in terms of reproduction and other things, but sometimes they also have compensatory effects. And so this is these are the curves for AAK1, AAK2, and then a double mutant that was created for this purpose. And you can see that for AAK1, there's not very much delay at all. There's no delay, but there's a kurtosis and no skew. For the double mutant, there's a little bit of positive skew for the one that was starved. There's no delay, and there's a little bit of kurtosis. But for AAK2, for some reason, none of these rules apply. It seems like the ones that are starved kind of just wait until like day 4 or 5 to dump all their eggs and reproduce. So this is what they're doing here with this curve. And so you can see them comparatively. We have a little confusing, but these are all the starved variants. Basically, that the AAK2 variant behaves far differently than both the AAK1 and the double mutant. So it's really interesting about this AAK2. Not really sure what's going on there, but there's clear maybe that AAK2 is doing some sort of compensatory mechanism. And AAK1 is basically getting hit with a penalty for being starved. And so we have another question here. So these are the examples. So then this is the last slide. Thanks to our contributors, and I will mention like all the contributors that we have. These are not all the contributors, but this is our sort of all the people I wanted to thank that contributed a significant amount. You're welcome to become a contributor. We have our weekly meetings on YouTube. We have a lot of sponsors here that have contributed to our efforts over the years, organizational sponsors. And so then that's it. That would be the whole talk. And then, let's see, we had a question. How does this all line up with energy that you're using? So about the mutants, or the starvation examples. So starvation is like what happens in C. elegans larval development is they have these protective things for energy. So when they get starved, they're using energy, they're putting energy into like surviving the starvation. And so when they do that, they it comes at a cost, I think, later in life. It's not really clear how this works, but there's a molecular pathway that controls this. So when you starve it, it's hitting the embryo, it's hitting the larval worm in a certain way. So if you have this AAK1 mutation, it basically reduces your ability to respond to that stress and development later in life. So a lot of the worms die or they don't reproduce very much. But there can also be a compensatory effect. That is, if there's a starvation event in larval life, it can respond by reproducing a lot or having an enhanced response later in life. And so that's it basically, yeah.
There's data on starvation of, I'm not sure if it's people in Holland or Denmark in World War II, and the effects on their grandchildren.
Oh, right, yeah. Yeah. Oh, yeah, yeah. So they actually have those in C. elegans. They do epigenetic type, epigenetic inheritance. We actually talked about that in the group there several weeks ago. But this is like a little bit different. I mean, they look at like, for the effects of multiple generations, maybe 3 generations. And actually in C. elegans, they've established that that's a thing, that's a very strong effect, but only for a couple generations.
Okay.
So Steve had to leave early. But
okay.
So were you able to come up with your?
Well, I have closed 100 or so windows.
But I can try to present. Yeah.
Okay, your entire screen. Now, select window or screen. I got fewer selections. Screen 1, screen 2, screen 3. Let me try screen 1.
Okay.
It seems to be the right one.
Okay.
I see a picture of you guys. Okay.
All right. Oh, yeah, that'll work. I can see.
Okay. Good, that worked. Okay. Okay, now this work started with a rather strange observation. If you take oil droplets with a small amount of surfactant and cool them slowly, they become polygonal. Here are some of the shapes. Okay, let's see if I can get some more. Squares, rectangles, pentagons, hexagons. Okay? And when I looked at them, I realized that archaea, which are prokaryotes, they're about 40% of the prokaryotes on Earth, often are also polygonal. Let's see, there's a triangular one, here's a square one, here's a rectangular one, here's a pentagonal one. Okay? And also diatoms are also often very regular polygons. There's a triangular diatom. So these are eukaryotes now, of course. And you can see some similarities here. Here's a square one, Here's one that's sort of pentagonal. Here's one that's hexagonal. Okay? And here's one with 12. Oh no, that's number 12. 1, 2, 3, 1, 2, 3, 4, 5, 6, 7. 7-sided. Okay? So I put these together with the idea that, which is not popular, called Archaea first. And that is that the first living organisms on Earth were Archaea. Okay? And we're trying to figure out two things here, and that is, what is the, if one assumed that life started as oil droplets, which became alive later, in the form of polygonal archaea, what might the conditions be, and how on earth did this happen? Okay? Now, what I've been reading literature on the origin of life, and most people worry about the chemistry. And there's nothing on shape. They just assume a spherical droplet, and that's it. Let's see if I've got some pictures here. Let's see. Here's a standard model for the origin of life. You have a spherical lipid droplet, bilayer, and the other molecules are just sort of floating on the inside, and somehow this becomes a living organism. Okay? So the question is, if we assume that life started out as a polygonal flat shape, how on earth could, this could possibly have started with these shaped droplets, which are kind of weird. I mean, we're taught that an oil droplet in water is a sphere. And this is generally true, but if you cool it slowly, you get these flat polygons. Okay? So this is what I'm working on now. And, whoops, no, that's not what I want. Here. Oh, okay. So I've got a new theory on the shape, which seems to be working. And that is that the shape is, here are some polygons that are all the same area with different numbers of sides, 3 to 12. Okay? And the idea is that the energy is proportional. Okay, I'm missing something here. And that is that if you look at Archaea, you get a, let's see, find a picture here. Okay, the surface of Archaea and bacteria, by the way, and also our dear little coronavirus, have these S-layer proteins which are stuck in the membranes and they're considered to form an extra layer outside the cell. Okay? And these differ between different species, but they come in various configurations. And there's a little bit strange thing in that if you look at, let's see, if you look at the, okay, they come in various, the S-layer proteins can have various configurations on the surface. But if you look at a cell, you can see that sort of domains in it. In other words, regions where you get one structure and then maybe another structure. And the polygonal ones the edges don't seem to line up with these things at all. So I was a bit mystified by that and came up with, well, here's, this is kind of fun. This is a picture of shapes of bacteria, and here's a picture of just one species of Archaea scaled to fit that. Let me make it bigger so you can see it better. Okay, this is a culture, a pure culture. Look at all the different shapes you get. Okay, of one particular Archaea. Okay, you got triangles, and I don't know, okay, here's a Here's a rhombus over here. Okay, all kinds of shapes. Here's a quadrilateral. It's sort of flag-shaped. Okay, these things don't seem to have good control over shape or even size. Okay, so they're kind of weird. Now, I was playing around with different ideas. Here's a sheet of cellophane that's been crumpled, and you can see that it forms all sorts of sharp edges, even triangles here. Just take a sheet and crumble it, you salt, you see this effect. Okay? So, the notion I came up with is that these are energy configurations. They're flat, they're two-sided polygons, and the energy is proportional to the area. If we assign an area to the cell, and we assign a perimeter and count the number of corners, I came up with this formula for the total energy, where A, S, and C are free parameters. Okay? And what I've been doing is, let me show you something here. If I run this in a simulation, oh, okay, there we go. Let's open this one. Okay, let's see. I'm still working on this program. I'm learning mathematically, which is a bear.
Yeah.
But here's an example of one of these simulations. I plot the energy versus the number of sides in these polygons, and they sometimes form neat minima. So for this particular set of values, I would predict that you'd get mostly six-sided, some five-sided, and seven and eight, etc. Very few four-sided, and you won't get any triangles in this case. 2 represents a full circle. Okay, so the energy here on a four-sided one would be the same as if it were circular. But what this would suggest is that this would come up with the minimum would be 6, so the most common would be 6 in this case, and you'd get these others. Okay, and I've been, let's see, let me just give you a little flavor here for what we're trying to do. Let's see. Well, it's not in there. I have to find it just a second. Okay, so. Okay. There we go. Should have done this beforehand, but okay, here we go. So these are our great grandfathers, if I'm right. And what I want to show you is a table we've been building up. Okay, this, yeah, just put it. Okay, now what I'm doing here is getting data out of the literature. Bacteria have, prokaryotes have surfaces that are gram-negative or gram-positive, which means they stain positively or negatively to a certain stain. And then I get data on how much sodium they can take, minimum and maximum, or the optimum temperature they can take, the temperature range. Many of these are thermophiles, they live at high temperatures. And they can live at strange pHs. And I can record get the pH out of each paper in a shape. Okay, as reported in the literature, and as I see it in the picture. So if it's italicized here, For example, here's one which was reported as irregular foci, but if you look at the pictures, you see quadrilaterals, hexagons, and heptagons, except the authors don't see that for some strange reason. Okay?
Yeah.
Now, I've done that for about 500 different species of Archaea. All of the files and some of them with antigens. Okay, and Natalie and I are now doing a statistical analysis of this to see under what conditions, if there's any pattern to what conditions you get polygons in. Okay?
Yeah.
And if we can find any pattern that might be indicative of the conditions under which these things originally formed. Okay?
Yeah.
So, and this theory might hold up. It's very simple. Basically, it's saying that the energy is primarily in those S-layer proteins, and the energy is different if they're or if they're stuck at the perimeter, or if they're stuck in a corner. But you get different energies for those three cases. Okay?
Yeah.
And what this suggests then is that people could look to see if the configuration of the S-layer proteins are, as one might expect, different at an edge than at a corner. And you might be able to measure the energy of those differences. So there's no literature on that, but this is sort of a prediction of what to look for. Okay?
Yeah.
So I've got a bee in my bonnet, I guess, which is to trying to see if shape is important in order to life, and if we can get these shapes, how on earth do other molecules interact with these shapes? That's the next question, because these are what are called confined droplets. Let's see, if I go back. Let's see. I'll go back to an original paper. This one. See, here are examples of molecules confined to droplets, and you can see the droplets become pentagonal, or become polygonal. Okay?
Yeah.
And there's one other thing, something called double droplet, which is basically water oil with a water droplet inside. And those might be precursors to real cells. If we have pictures. No. Okay, so that's where I'm at.
Wow, that looks pretty. Oh, there's a Wurfel. Yeah.
There's two Wurfels in here for some reason.
Yeah. Well, that's very good. Yeah.
Okay, so that's kind of where things are at. We're trying to puzzle out under what conditions archaea become polygonal, and then there'll be other problems like looking to see if the ones that are polygonal have any, maybe older. I don't know. And see, there's all kinds of weird things here. If you take a sphere, basically, what I've come up with is two ideas here. Why would you want to be flat? Well, a flat cell will, at the origin of life, a flat droplet would concentrate molecules that are already inside it, and concentration is key to the chemistry of the origin of life. Okay?
Yeah.
And another thing is that it becomes essentially two-dimensional instead of three-dimensional, so the molecules, how should I say, they can move around in 2 dimensions better than they can in 3 dimensions. And this is an old phenomenon. It was discovered back in the 60s, having to do with moths. You know this business about how moths and butterflies can detect each other with pheromones in enormous distances? And that's apparently because the molecule gets on the antenna and then can wander along the antenna to a receptor. So the idea of dimensionality reduction might also be key here. Okay?
Okay, yeah.
Okay, so that's where we're at.
Okay. Thanks a lot. Yeah, it looks really interesting. Is there any questions by anyone?
Yeah, how do I stop presenting?
Oh, it should be in the lower right-hand corner. It should.
Lower right-hand corner. Nope, nothing's coming up there. My print layout. Nope, there's nothing here.
Oh, well, let's see.
Let's see.
Yeah,
What am I doing here? Control sharing. Ah, who on earth is that? Ah, there.
All right. There we go.
There. Okay. Oh, stop presenting. There we go.
Yeah, there we go.
Okay.
All right.
So it's kind of fun. The title of the paper is "When We Were All Triangles."
Is this something you're doing for a special issue, or is it going to come out?
Well, I'm going to try to get it in the book. If we can finish it this month, we'll send it in for the book, Conflicting Origin of Life Models.
Yeah.
Okay? But it's kind of fun. It's weird that shape has been totally disregarded by the chemistry community, which thinks they have the key to origin of life. But they can't find it.
Yeah.
Okay, so that's where it's at.
That sounds good. Yeah. Oh, sounds like we have to go.
Okay. Oh, somebody's calling me on Skype.
Okay. Well, let's wrap up the meeting. We're past the top of the hour. And, so next week we'll have the Google Summer of Code students. They'll be announced today. And hopefully they can come and to the meeting and introduce themselves, and we can discuss and interrogate them a little bit about like what they are going to do this summer and how that might fit into a lot of the data that we have and outstanding questions. So make sure you bring your questions, and they'll want to make them feel welcome. So okay. All right, everyone.
Okay.
See you later.
Okay, bye-bye.
All right, bye.